Hi everyone, we will do lab exam 2 exercise today. This lab exam number 2 will be over lab experiments 7, 8, 9, 10, 11, 12, 13, 14, and 15. For the best results of these experiments, make sure you repeat lab experiment 7 until 15, as many as possible before taking this exercise. Prepare some pieces of papers and pen or pencil to write your answer. There will be 2 or 3 seconds after each question to write your answer on paper before the answer key is given and explained. Remember, you can always pause the video if you need more time. After finish the exercise, check your answer, how many are correct and how many are incorrect. Repeat the process until you get 100% correct answers. Make sure that the 100 correct answers are based on your knowledge, not from guessing. It is very likely that you will get a very good score on your lab exam number 2 if you follow this instruction correctly. Now we start with number 1. Which structure is not trimmed away in an eye dissection? So during the eye dissection, which part of this eye will not be trimmed away? A, B, C, or D? Okay, look at this picture. Okay, so there are a lot of muscle fats that have to be trimmed away before you perform the eye dissection. So the muscles, the eyelids, if still there, then you have to remove it in order to perform good dissection. Fat tissue also have to be removed or trimmed away. The one that stays is the optic nerve, which is this one over here. Okay, so optic nerve is not trimmed away. The optic nerve or cranial nerve number 2 carries the special sense of hearing, tasting, balance, or vision. So the name is telling you is optic. Optic means vision. So it carries the sense of visions from our eyes into our brain. So that's the correct answer. Where do you perform a coronal cut in the eye dissections? So coronal cut means you cut organs or part of the body that will divide into front and the back part. So when you perform the eye dissections, which one that you have to do? A, B, C, or D? Okay, through the iris is not, this is very front part. Through the sclera, just posterior to the cornea. Yes, this should be the answer. If you look at this picture, so this is the cornea, okay, and this is the sclera, which is the tough layer of eyeball. So this is when you cut this eyeball round, and then it will split the eyeball into front part and the back part. This is what we call the coronal or frontal cut. So through the sclera, just posterior to the cornea. Which of the following structure is not considered an accessory structure? So it's not. Okay, so when you see the eyes, look at the eyes over here. Okay, the main structure that used for vision is actually the eyeball. So eyeball is the main structure for vision. The rest of the structure will be called the accessory structure. Okay, like for example, this lacrimal glands or tear glands, the eyebrow, the eyelashes. Okay, all of them is accessory structure. So which one is not? Okay, the eyelids will be accessory structure because you still be able to see if you don't have the eyelids. Okay, the lens, the lens will be the main structure. So this should be the answer because there's extrinsic muscle, also the accessory structure. Lacrimal gland is also accessory structure. So the one that is actually the main structure for vision is the lens, which is part of the eyeball. So lens should be the answer. Which of the following accessory structure of the eyes move the eyeball voluntarily? So you can see to the left, to the right, up, bottom is because of this accessory structure. So what is the name of that accessory structure that move the eyeball? Of course, eyelid is not, eyelashes will not move your eyeball. It's going to be muscle. Okay, so muscle is the one that contract and move the eyeball. That muscle is called the extrinsic muscle because that muscle actually outside the eyeball. There are 6 of them. Okay, 6 extrinsic muscle that move the eyeball. The lacrimal gland is produce the tear. It's not really move the eyeball. So the answer will be the extrinsic muscle, which is 6 pairs. Okay, 6 muscles on the left eyes, another 6 muscle on the right eyes. Okay, so while reading, we move our eyes along the text from the left side page to the right side. So from the left to the right. Which extrinsic eye muscles are needed for this process? A, B, C, or D? Now, to answer this question, you have to know, you have to learn the name of these 6 muscles on each eye. So this is the right eyes over here. There are 6 muscles. The top one is called the superior rectus. The bottom one is called the inferior rectus. The one that close to the nose is called the medial rectus. The one that is away from the nose is called the lateral rectus. So these 4 muscles are straight. It's called rectus. So rectus means straight. There is another 2 muscles that is diagonal or oblique. One on the top is called the superior oblique, and the bottom one is called the inferior oblique muscle. Okay, so these 6 muscles for each eyeball. Now, if we move our eyeballs from left to the right, so left to the right. So on the left eyes, this medial rectus should be the one that is contract. For the right eyes, the one that contracts should be the lateral rectus muscle. So look for that one. Medial rectus for the left eyes. Okay, so medial rectus of the left eye and the lateral rectus of the right eyes. So that should be the answer for that. But when you move from the left to the right, this is for the left eyes. Okay, the medial that will be contract. For the right eyes, the lateral muscle, that the one that will be contracted. Okay, so that's the answer. If you move your both eyeball up, then the superior rectus for both eyes, left and right, will be the one that contracted. Okay, so that makes sense. Okay. Now, when a bright light enters the eyes, the blank muscle of the iris will contract, causing the pupil to blank. A, B, C, or D? Okay, so when the bright light enters to the eyes, then the pupils will be smaller. It will be smaller. It means that the eyes is actually constricted. So if you see this picture over here, so this is when there are a lot of light enter. This is during normal light, okay? And this one is when low light conditions, the pupil is become bigger or wider, okay? So for this question is during the bright light. So which muscle will be contracted? It's going to be this muscle over here. Okay, it's called the sphincter muscle. So the sphincter muscle will be contracted. Sphincter, okay, will be contracted and it will constrict the muscle. The iris will contract, causing the pupil to be constricted. Okay, so that should be the answer. Okay, the pupil will be smaller, okay, because of the constrictions of the sphincter muscle. In which condition your eyes pupil will be fully dilated? In what condition? A, B, C, or D? Of course, we already mentioned that in the low light conditions, you need to open your eyes to dilate the eyes, the pupil. Okay, so in low light condition, the pupil become bigger. So this is when the pupil is wider. So this is usually when during the low light condition, for example, during the night. The space between the cornea and the lens is filled with, okay, if you see, this is eyeballs. So you have cornea in the front, and there's a lens over here. Okay, so lens is actually divided the eyeball into front part and the back part. Okay, so between this lens and cornea, it's filled with liquid. And between the lens to the retina, so retina is on the back, there is also filled with the liquid. The liquid that fills between cornea and the lens is called the aqueous humor. The liquid that fills the lens and retina is called the vitreous humor. Now, if your question is the space between cornea and lens, okay, cornea and lens, which is this area, filled with the aqueous humor. Which eye structure support the retina to keep it flush against the back of the eyes? So again, this is the eyeballs. Okay, this is cornea. You have lens. Okay, and between the lens and retina, there is a liquid that actually support the retina. So that liquid is called the vitreous humor. So that should be the answer. The first nerve cells light will encounter when it's reached the retina are called rod, cones, bipolar cell, or germline cell. Okay, so again, if you look at this eye structure, like this, you have cornea, lens, and then retina. Okay, so the light will enter from this. Okay, cornea go to the lens and finally reach to the retina. In the retina, it has 3 layers of nerve cell. Okay, the very back layer is actually the photoreceptor, which is rod and cone. Rod and cone. That's a very back. And then between this, in the middle, there is bipolar layer. So the second layer is called the bipolar layer. And on the top over here, there is another layer of nerve cell. It's called the ganglion layer. Okay. So from this information, then you should be able to know when the light enters to the retina, to the eyeball, the first one that will get the light is actually the ganglion. Okay, the ganglion cell. Visual acuity is measured using A, B, C, or D. Okay, so when you go to the eye doctor, you're going to use something to measure your visual acuity, whether you are normal, which is 20/20, or maybe less than normal. Okay, so what the doctor use is using chart. It's called the Snellen eye chart. Okay, it's look like this. Okay, with letters with different size. If a person see the Snellen chart just like this from 20 feet, so from the 20 feet, and he able to see the 20 size of letter, which following condition could be excluded. So if he able to see the chart from the 20 feet, it means that he is in normal vision. Okay, so the one that should be excluded is the myopia. What is myopia? Myopia is nearsightedness. So if you have nearsightedness, you will not be able to see this chart from 20 feet. You have to go closer, because you only see closer object. Okay, so this one has to be excluded. So if you're able to see 20 feet, this Snellen chart, it means that you are normal. So the myopia will be excluded from this diagnosis. Stigmatism is assessed using A, B, C, or D. Okay, of course, we already used this Snellen chart for visual acuity, so it will not be used for stigmatism. It's going to be used another chart. It's called the stigmatism chart. It's look like this. So if you have stigmatism, you see this three line. If you have stigmatism, this three line is become only one dark or thick line. Maybe number 2 and number 8 over here. 1 and 7. So you don't see the three line, but you only see one big or thick line. Okay, so that's the indication of stigmatism, and it's measured using this stigmatism chart. Stigmatism is a result from a defect in the A, B, C, or D. This is mostly caused by the cornea, irregular cornea. The cornea is not soft, but it irregular shape. It will causing this stigmatism. Stigmatism is mostly caused by problem with, I just mentioned that, the shape of the cornea. Okay, so if someone have a normal cornea, this letter A will be really clear. You can see this letter A very clear. But if the cornea is irregular, is not soft, then what happens? This letter A is become kind of blurry or even become thicker. unable to read a clock with when it is 11 o'clock, when it is 11 o'clock, but reading is fine with other times or numbers. Maybe you can see 2, 3, 9 easily, but you cannot see number 11. Number 11 become thick, just one line, but thick. So this is a sign of, okay, just remember, this is the sign of stigmatism. So stigmatism cannot see double lines or triple lines. So these lines just become one thick line. So if you see this number 11 over here, it has two lines, 1 and 1, right? So if someone has stigmatism, this 11 is become just thick number 1. Okay, that's an indication of stigmatism. Okay, over here, okay, so number 11. Blind spot is defined as the area A, B, C, or D. So blind spot is the area in the retina. So remember, retina is the place for the photoreceptors. Okay. So the retina, there is a part of retina that do not have photoreceptor. That area is called the blind spot. Okay. So the retina, where the nerve fiber leave the eyes and form the optic nerve. In this area, no photoreceptor. Okay, because this area filled with the nerve fiber. This is going to be the blind spot over here. It's called the blind spot or optic disc. Okay, that's the same name. In this area, there will be no photoreceptor. Therefore, it's going to be blind spot area. You cannot see anything if the object go to that blind spot. The blind spot does not have any blank and therefore produce no visual image. A, B, C, or D. So it doesn't have any photoreceptor. Okay, and there are two types of photoreceptor. From black and white vision, the photoreceptor is called rods. For the color vision, the photoreceptor is called the cones. Okay, it's based on the shape. So rod photoreceptor look like rod or stick, and the cone photoreceptor is look like an ice cone, like this. Okay, so these are the two photoreceptors. Now, look at the answer. This is not correct. This is yes, and of course, both of these. So the answer will be there is no rods and cones in the blind spot. What are rod cells? A, B, C, or D? Remember, the rod cell is the photoreceptor that used for black and white vision. So this is the one that detects the amounts of light but no color. So this is the clue over here. So no color vision for rod cell. Normal type of vision or 20/20 is called A, B, C, or D. Okay, so this is the memorization. You have to memorize the scientific name for normal vision. Yeah, normal vision is hematocrit. So this is terminology for normal vision. Myopia is nearsightedness. Okay, so this person only able to see close object. Hyperopia is opposite. This is far-sightedness. So this individual only see the farther object. Nyctalopia is for someone that unable to see in low light condition. In low light condition. So this is the terminology for this one, nyctalopia. So normal vision is emmetropia. Okay, emmetropia is normal vision. Nearsightedness vision is called, we just mentioned that, we just explained it, it is myopia. So myopia is nearsightedness. A person with longer eyeballs is said to have myopia or nearsightedness. This means the image held further away from the eye appear blurry because the points of the eye's focus is blank of the retina, A, B, C, or A, B, or C. Okay, so because this person has longer eyes, longer eyeballs, then the focus is actually fall in front of the retina. It is not on the retina. It is in the front of the retina. So that person has to go closer in order to see an object in focus. So the answer will be in front of the retina. Okay. It's over here. Farsightedness vision is called, and this is terminology, memorization, is called the hyperopia. Okay, so this is the farsightedness. So this individual actually have shorter eyeball. Therefore, the object, the focus is carried behind the retina. So he has to move backward in order to see object in focus. Difficulty seeing in low light condition is called, and this is terminology for nyctalopia, and this is usually caused by lack of vitamin A. Okay, so someone that do not get enough vitamin A, they usually have problem to see during the low light condition, during the night, for example. Condition of older people when the lens decrease its ability to accommodate near vision is called A, B, C, or D. Okay, just remember older people, when they read the newspaper, they have to move that newspaper back. This is because their accommodation is not good anymore. Okay, it's not elastic, so it's changed, it's decreased. So this condition, again, this is terminology, memorization, is called the presbyopia. This is older eyes, presbyopia. The near points of accommodations, what happens when someone become older, A, B, C, or D. The near points accommodations increase or decrease. So near point is the object that you can see, focus, okay, closer as your to your eyes. So when we are older, this near point is actually increased, become farther away. So it will increase with age. So usually normal near point for younger people is around maybe 6 inches. But older people, the near point may be 12 or 16 inches. So he has to move the newspaper away in order to read the letters. So that's what we call the near point. Okay, so when we see the close object, our lens is actually become shorter, is bulging. Okay? When we see the far objects, the lens is flatter, flat. Okay, so when we are older, we lost this ability to make our lens shorter. Therefore, our near point is going to be here. Okay, because this lens is not bulging. Okay, so that's what we call the presbyopia, older eyes. Does the elasticity of the lens increase, decrease, or remain the same with age? A, B, or C? So already mentioned, it will change. Okay, the elasticity will be decrease with age. The near point increase with age. Okay, just remember the difference between elasticity and the near point. The elasticity is actually for the lens accommodation. It will decrease with age. So we actually unable to really perform the lens accommodations when we are older. Okay, so this is elasticity will decrease with age. To measure hematocrit, now we are in blood experiment. To measure hematocrit, blood sample must be blank, A, B, C, or D. Okay, remember when you perform this lab experiment, you take the blood samples and we centrifuge the blood. Okay? So we put in the tube, we centrifuge the tube, and then after the centrifugations, then you will have 3 layers in the tube. Okay? The bottom layer will be darker because this layer has the red blood cell. Okay? The middle layer is buffy coat. It consists of some fat and white blood cell. And the top layer will be clear because this is actually the blood plasma, water. So this, when you get the hematocrit, okay, you have to centrifuge the blood sample. After centrifugations, the blood sample will produce blank layers of blood part. We just mentioned that. How many layers? There will be 3 layers. Okay, 1, 2, 3 layers. When a blood sample centrifuged, the top layer represents A, B, C, or D. Okay, the top layer will be clear, and this actually the blood plasma percentage. And normally it's going to be 55% for normal or healthy blood sample. When a blood sample centrifuged, the buffy coat in the middle layer represents so the 3 layer again. The top layer is plasma. We already mentioned that is 55%. And the middle layer is consists of white blood cell and platelet percentage. Okay, so this middle layer of here, maybe about 1% or less than 1%. when the blood sample centrifused, the bottom layer represents the, and this one, the top layer, 55% is the blood plasma. The middle layer is maybe around 1% or less is the white blood cell. Okay, and the bottom layer, about 45%, is the red blood cell. So this should be the answer. This is when we measure the hematocrit, is actually measure the percentage of the red blood cell. Okay, so about 45% for normal or healthy blood. The most abundant form elements in the blood is the, okay, so blood actually has 3 types of cell or form elements. The first one is called the red blood cell, about 99%. Second one is platelets, okay, maybe 1%. And the third one is white blood cell, less than 1%. Okay, so that's the percentage of these form elements or cell. Okay, so which one is the most abundant one? Will be the red blood cell. And the scientific name for red blood cell is the erythrocyte. So erythrocyte is red blood cell. Leuco means white, so this is white blood cell. Thrombo means platelets. Okay, that's the scientific name. So the answer will be erythrocytes or red blood cell. Hematocrit or HT is a measurement of the percentage of blank in the blood. So again, this is the bottom layer of the centrifugation, about 45%. This is red blood cell. Okay, so measure the red blood cell or erythrocyte. Hematocrits of blank would be about normal. So I mentioned this several times. For normal, healthy blood cell, the hematocrit is around 45%. Okay, so 45% is the healthy hematocrit. Oxygen transport in the blood is provided by the A, B, C, or D. Oxygen is transported inside the red blood cell. There is a protein, it's called the hemoglobin or HB inside the red blood cell. So it is transported by the erythrocyte or red blood cell. If a person has hematocrits lower than 38%, okay, so normally is 45%, it is even lower than 38%, he might be has abnormal blood condition called A, B, C, or D. Okay, remember hematocrits measure the red blood cell concentration, and red blood cell carry oxygen. Okay, so someone that have lower hematocrit, it means that he has less oxygen carry in the blood. And the condition when someone have less oxygen is called anemia. And the sign and symptom usually they become weaker, okay, sleepier because of this less oxygen in the blood. So this is anemia. If a person has higher than 60% hematocrit, he might be has abnormal blood condition is called. So it means that the red blood cells is so many in the blood. Okay, the blood become stickier. Okay, because less water and more red blood cell. So this condition is called the polycythemia. So polycythemia is condition where the red blood cell is higher or the hematocrit is higher than normal. Which type of white blood cell is this? So this is part of your lab experiment. When you see the blood sample under the microscope, you should be able to identify 5 different type of white blood cell. Okay, so there are 5 different type of white blood cell. The first one, the most abundant one, is called the neutrophil. Okay, and neutrophil usually have many lobes in the nucleus. So one nucleus with many lobes. So, and the second one, the second abundant one is called the lymphocyte. Lymphocyte is going to be easy to see. It's smaller. And then you should be able to see big or large nucleus. So almost all the cell fill with the nucleus. So this is going to be lymphocyte. And nucleus usually blue in color or purple in color under the microscope. The third one is called the monocyte. Okay, monocyte is really big type of white blood cell. And usually you should be able to see the nucleus look like kidney. So kidney-shaped nucleus. So this is monocyte. Number 4 is eosinophil. Okay, so eosinophil, you should be able to see cell with red, okay, red granules or crystal. Okay, and the last one is called basophil. If you see under microscope, you're gonna see blue granules. And you don't really see the nucleus because the nucleus color is blue. It will be covered with this granule. For the eosinophil, you still be able to see the nucleus is look like earphone. Okay, look like an earphone. That's gonna be eosinophil. So just remember this. Remember this characteristic of this white blood cell. Now we go back to this question. Look at this picture. This white blood cell over here, it has nucleus with many lobes. Okay, so many lobes. So which one's going to be? Of course, going to be number 1 over here, the neutrophil. So the answer will be neutrophil. Okay, look at this one. Which one is this? This is red granules with the nucleus look like an earphone. Okay, so which one is that? It's gonna be the eosinophil. Okay, look at this one. So this white blood cell is covered with the blue granules. You don't see the nucleus over here. So which one is that? This is the basophil. Okay, look at this one. This white blood cell is small and nucleus is big. The whole cell is kind of covered by this nucleus. So which one is this? This is the lymphocyte. Okay, so it's easy actually to identify these 5 different types of white blood cell under the microscope. Okay, make sure you know how to do it. Remember that. Okay, try to remember shape and structure of this white blood cell. Now, the last one. The nucleus look like a kidney shape, okay, and it's really big white blood cell. So which one's gonna be? You have to memorize it. This is the monocyte. Okay, monocyte has a nucleus look like a kidney shape. Okay, now the most abundant white blood cells are the A, B, C, or D. Remember I mentioned before, the most abundant one is start with N. The second one is start with L. The third one is start with M. And the next one start with E. And the last one start with B. So the most abundant one is neutrophil, and then lymphocyte, monocyte, eosinophil, basophil. How to memorize that? I usually use mnemonic. Never let monkey eat banana. Okay, so never is N. That's the most abundant one, which is the neutrophil. The least abundant white blood cell are the A, B, C, or D. Remember, never let monkey eat banana. So the least abundant one will be start with B. Never let monkey eat banana. It's gonna be basophils. The white blood cell that are greatest nuclear variation is the, so this is the one that has nucleus with many lobes. So which one is that? Remember, that's the neutrophils. The white blood cell that has reddish red, okay, reddish granule, is the eosinophil. Eosin means red. Okay, so eosinophil has a reddish granule. And the nucleus look like an earphone. Okay, remember that. The white blood cell that has bluish stains granule is the, okay, so this one has blue granules that call for the nucleus. So you don't see the nucleus in this white blood cell. So baso means blue. So just start B also. So blue basophil. What does a different white blood cell count? Different white blood cell count. What is this? It is a total number? Of course it's not. Okay. The total number of white blood cell, it is not. Okay. The percentage of different type of white blood cell as a fraction of the whole white blood cell. Yes. Okay. So it should be the answer over here. The antigens related to the ABO groups are located A, B, C, or D. Now we are blood typing. Okay, so there are 4 different types of blood based on the ABO blood type. Okay, so type A blood, and then B, AB, and O. So this different type of blood type is actually based on the antigens. And these antigens located on the red blood cell. So this red blood cell carry the antigens. If the red blood cell carry antigen A, it will look like this. Okay. Look at triangle, antigen A. Then that person will have type A blood type. If the red blood cell carry antigens B, let's say it look like round over here. So antigens B, then that person will have type B blood type. Now, if the red blood cell carry both antigens, A, B, and also A, like this, then this is B antigen, this is A antigen, then its blood type is going to be combination, which is AB. So AB blood type because it's carry both antigens. Now, how about O? O blood type, it does not have the antigen. Okay, so his blood will be O type. So the antigen is located where? Is on the red blood cell. It's a protein that is carried by the red blood cell. The antibodies related to ABO blood groups are located where? So remember our blood, okay, is actually consists of plasma, which is water, okay, it's watery part, and the cell, okay, and the most abundant cell is red blood cell, then platelets, and the white blood cell. So blood types will be related to the antigens carried by this red blood cell. So red blood cell carry the antigen. In addition of the antigens, our body also produce what we call the antibodies. And these antibodies is actually located in the, or produced in the plasma. Okay, so it's going to be in the plasma. So this is the rule. If our blood type is A, it means that we have the antigen A. Okay, so antigen A in the red blood cell. Then we will be able to produce antibody. The antibody will not A, okay, because if antibody A, it will destroy the antigen A. So in our plasma, we will have antibody B. Okay, so it's going to be round like this. If your blood type is B, which is carry antigens B, then you will be able to produce antibody A only. Okay, if your blood type is AB, then what happens? You will not produce antibody. No antibody. Because if you produce antibody A, it will destroy this A antigen over here, and your blood will be destroyed. If your blood type is O, it means that you don't have antigen, no antigens, then your body, your plasma will able to produce both antibodies A and B. Okay, this is antibody A and antibody B. Okay, so that's the rule. Okay, so the antibodies will be produced in the plasma. The surface of red blood cell of a person with type B blood has, and so just follow the rule over here. If you are type B, then your antigens will be B antigen. That's it. And so your red blood cell carry antigen B. So blood type is based on the antigens, not based on the antibody. Okay. The surface of red blood cell, a person with type AB blood has what? Of course, because you have A and B, okay, then it's going to be you have antigen A and B, both antigen A and B. Okay, so your red blood cells will carry The surface of red blood cells of a person with type A blood will carry what antigen? So red blood cell, if you are type A, then you only carry antigen A, which is this one. The surface of red blood cell of a person with O blood type, so O, with antigens you carry? None. None or neither A nor B antigens. No antigens on people with blood type O. In the plasma of a person with type A blood has what? Antibodies. Remember, this person is type A. So it has antigens A. Okay? If you have antigen A, then you will not produce antibody A. You will have or produce antibody B. That's the rule. So if you are type A, then the antibodies that you produce will be antibody B. In the plasma of a person with type B, so this one have B antigen. Okay, then you will produce what type of antibody? Again, the rule, the antibodies that you will produce gonna be opposite, not B, but gonna be A. In the plasma person with type AB blood, so you carry both antigen A and antigens B. So what type of antibodies you will produce? Because you have antigen A, you will not produce antibody A. And because you have antigen B, you will not produce antibody B. To mean that no antibody. Okay, so neither A or B antibodies. In the plasma of a person with type O, okay, so type O is look like this, no antigen. So what antibodies you produce? Because you don't have antigens, then you will be able to produce both antibody A and antibody B. Okay. If the blood from individual with type A blood mixed with the antiserum A, so antiserum is the same as antibody in the first well, and with antiserum B in the second well, what would be the result? So you are type A. It means that your blood has antigen A. Okay, so your antigen A, if you mix with the antiserum A, then your blood will be broken down or agglutinated. Okay, because antibody A will destroy the antigen A. Okay, the antiserum B or the antibody B will not destroy the antigen A. Okay, so when you blood typing your blood and you mix with the antiserum A in the first well will show the agglutination, which is broken down. Okay, and the second well will not. So that should be the answer over here. Okay, so the first well will be showing the agglutinations, the second well will not. Okay, if the blood from type B, okay, type B, it means that you have this antigen B. Okay, the first well, it has antibody A. It means that your blood will be okay. There is no agglutination, it's not broken down. And the anti-B, on the the second well, then there will be agglutination. So look for that one. The first well show agglutination. No, the first well will not show agglutination, right? The second well will, yes. So this will be the answer. Okay, so again, remember this activity, you do it or you did it at the lab experiment, the virtual lab. If the blood from an individual with AB, so AB means you carry the antigens A, okay, and antigens B. Okay, now what happened with the antibody or with antigen? In the first well, you have antiserum A. It means that it will destroy this blood. Okay, the blood will be agglutinated. And the second well, there is antiserum B. This antiserum B or antibody B will also destroy this antigen B. So you will also see the agglutination or destruction of blood in the second well. So look at for that one. Both first and second well will show agglutination. That should be the answer. Now, if your blood type is type O, okay, no antigens, then what happens with when you put the antiserum on the first well and antiserum B on the second well? Both of them will not show agglutination. The blood will not broken down. So neither first and second well will show the agglutination. So the blood is stay fresh, no agglutination. The A, B, and RH antigens are located. Just remember antigens where it is located. It is located on the red blood cell. Okay, so if we put all the antigens, antigen A will be looked like this, antigen B look like this, maybe RH antigen look like square. Okay, so all of them are proteins that are carried on the red blood cell. The anti-A, anti-B, and anti-RH antibodies are where? Again, antibodies are produced in the plasma. Okay, it's dissolved in plasma. Which type of blood has all type of antigens on red blood cell and no antibodies in the plasma? So antigen, this means that this blood has all of them. Antigen A, okay, antigen B, and antigen rhesus. So because they all have antigens A, B, and rhesus, then there will be no antibodies produced. So which one do you think? So for the one that carry rhesus antigen is called plus. Okay, is blood type positive. So which one carry this all antigen? It's gonna be, it has A, it has B, and it has rhesus, which is positive. So this should be the answer. Which type of blood has A, so A antigen, and rhesus antigens on the red blood cell? Okay, so A and rhesus will be positive. So just look for that one. Okay, don't worry about this antibody. So this blood type will be A positive. Which type of blood has B and rhesus? So just look at that one. So it has antigen B and antigen rhesus, B and plus. Right? That's how to recognize the blood type. Just look at the antigens. So B plus, which is this one, okay, B positive. Which Which type of blood has B antigens? So it's going to be B. And no rhesus antigen. So it's going to be negative. So B negative. Which type of blood has no antigens? So no antigens is mean that this is O, right? And all type of antibody. So it's O and also negative. It's gonna be O negative. Which type blood has B antigen? So it is B and no RH, rhesus antigen. So B negative. B negative, this one. Okay, now this is the experiments or blood typing that we perform in the lab. The following results of blood typing show what blood type. Look at the A. So A, no agglutination. It means that there is no antigens A. No antigens A. Okay. B, okay, there is agglutination. So it means that it has antigens B, okay, because it's agglutinated with the B antibodies. The rhesus over here is also agglutination, okay. It means that it has rhesus antigen or positive, okay. So you combine this with blood type over here, okay. It has B and positive. So that's going to be the blood type B positive. John Doe, who has AB blood type, has an accident and is required to have fast blood transfusion. The hospital does not have AB blood type, but it has A, B, and O blood type. Which blood type should be used for the transfusion? Remember, for the transfusion, if you are AB, it is better to have donor that is AB2. Okay, so AB and AB is the best. But if you don't have the similar blood type, then O can be used because O is considered as the universal donor. Why it is universal donor? Remember, O, it does not carry any antigen. So if you receive blood O, it will not destroy or it will not agglutinate in your body because there is no antigen. Even though you produce the antibodies, there is nothing happening. Okay, O is the universal donor. So it should use the O blood type if there is no AB for this condition. Jimmy Neutron has AB blood type. Okay, which blood type should he receive transfusion? Of course, the first one, use AB if AB available because that's the best matching. Okay, if not, then use O. Okay, so look at that. Type AB blood. Okay, in emergency, type O blood. Yes, this should be the answer. Use AB first. If not, then you can use O blood type. Which of the following arteries is the most common use for palpation? Now, your experiments with measuring the heart rate with the palpation. Which one is the common one used for palpation to measure the heart rate? It is on your hand, on your arm, which is the radial artery. Okay, so if it's your hands like this, radial artery is over here. So this is the most common area for palpation to measure the heart rate. Which of the following arteries can be palpated on the surface of the body to measure pulse rate? Vertebrae, renal. Of course, renal is inside, you cannot use it. Okay, common iliac is also inside your body. It's going to be very difficult. Vertebrae is the same. So this will be the one that close to the surface area is dorsalis pedis artery. It is on your foot. Which of the following arteries can be palpated in the inguinal regions to measure pulse rates? So this is in the inguinal of our area over here. Okay, so what is the name of this artery? Femoral, internal, common iliac, or popliteal artery? This is the femoral artery. Okay. What do you measure when determining blood pressure? So blood pressure is measured which pressure? Vein? Of course not. Okay, vein is a very low pressure. Arterial? Yes. So this is actually measuring the arteries pressure. Which of the following arteries is used for determining blood pressure? Okay. Which of the following arteries is used for determining blood pressure? It's a brachial artery. So when you go to the doctor, the nurse will take the blood pressure using this brachial artery on the upper arms. The stethoscope is placed over blank when taking the blood pressure. We just mentioned that on the brachial artery. During ventricle contractions, the arterial pressure reaches the maximum, which is called what? So there are two cycles. The top one is called the systole. This is when the pressure is highest. And the second cycle is called the diastole. This is when the pressure is the lowest. Okay, so the maximum pressure will be measuring the systolic pressure. And usually, normally is 120. And the diastoles normally 80. So 120 over 80. That's the systole and diastole. When the ventricle relax, okay, and then the pressure will drop to the lowest. And it is measured what? We just measure it. This is the diastole. Okay? The lowest pressure is the diastolic pressure. Blood pressure is expressed as a fraction. The top number of this fraction indicate the, let's say, 120 over 80. So the top number is what? It is the systolic pressure. Okay. Now how about the bottom number? The 80 over here. This is measured the diastolic pressure. The first cross sound occurs when the blood pressure is equal to the, so the first sound is actually when the pressure is the highest one. So which one is that? It is the systolic pressure. Let's say 120. And the last one, the last cross sound occurs when the blood pressure is equal to the lowest one, which is the diastolic pressure, maybe 80. Which of the following resting blood pressure would be considered normal? 80/55 is too low. It's not. 90/60, it is still low. Okay, it is not. 120/80, yes, this is normal. This 140/90, it is hypertensive. So this is not. So normally 122/80. Which of the following resting blood pressure would be considered hypertensive? Of course, it's over the normal range, which is going to be 140/90 is considered as hypertension. Why did the pulse rate decrease 5 minutes after exercise? Again, this is virtual lab that you perform before exercise. After exercise, the pulse rate will be increased. And then after that, 5 minutes after exercise, the pulse rate will be decreased again. Why? This is because the tissue demands for oxygen has decreased, okay, after 5 minutes resting after the exercise. That's what you get when you perform that experiment in the lab, a virtual lab. When a person moves from sitting position to laying position, his blood pressure decreases. Why? A, B, C, or D. Again, this is the experiment that you did on the virtual lab. So the blood does not have to travel against gravity when we are laying down. So the pressure is decreasing. When a person moves from sitting position to standing position, his blood pressure increases. Why? Again, this is the experiment that we perform because more skeletal muscle are active and it requires more blood. So your heart will pump more blood, the blood pressure will be increased. How many chambers does the heart consist of? So this is about the structure of the heart. So the heart has 4 chambers, 2 on the top and 2 at the bottom. Between the top chamber, the 2 on the top and the bottom, it has connection. It has valve. Okay, so 4 chamber. The top one is called the atrium. The lower chamber is called the ventricle. So 4 chamber. The top chamber of the heart is called, we just mentioned it, the top chamber is called the atria or atrium if it is only one. The bottom chamber of the heart is called, we just mentioned that, ventricles. That's the bottom chamber of the heart. Which chamber receives freshly oxygenated or rich blood from the lung? A, B, C, or D. So just remember, if we talk about receive, it means the atrium. So just look at the atrium. So this is the right side, right atrium. This is the left side, the left atrium, connected to the left ventricle. This one connected to the right ventricle. Okay, so the lungs will put the oxygenated blood to the left atrium. Left atrium will have a lot of oxygen because it receives blood from the lung. Okay, so right lung and left lung over here. So it's gonna be the left side. So the left side of our body or of the heart over here, it will have rich blood. Okay, or blood that has a lot of oxygen, oxygenated blood. So just remember that, the left side. Okay, the left side will have blood that has a lot of oxygen. So left atrium. Which chamber received the deoxygenated or poor blood from the body tissue? So after our tissue use the blood, oxygen will be gone because it's using for respiration, cellular respiration, and it will go to which one? So receive always atrium. So it's going to be on the right side. So the right side of our heart, so right atrium and right ventricle will have poor blood. Okay, the blood that do not have oxygens anymore. Which chamber pumps deoxygenated or poor blood through the pulmonary trunk to the lungs? Okay, remember again, if we talk about pump, this is going to be ventricles. Okay, and remember the right side is the one that has poor blood. Okay, so the one that pump the poor blood to the lungs will be from the right side, which is the right ventricle. Which chamber pumps the oxygenated or rich blood through the aorta to the body tissue? So our body tissue needs oxygen. It will be pumped from which part of the heart? Remember, when we talk about pump, it's going to be ventricle. And just remember again, okay, the left side of the heart is the one that has rich blood. Okay, so the blood will be pumped from this left ventricle, okay, on the left side. So left ventricles will be the one that pump the rich blood to the whole body tissues. What is the function of the 4 heart valves? A, B, C, or D? So when we talk about the heart chamber, okay, so you have right atrium, left atrium, right ventricle, left ventricle. Between right atrium and right ventricle, there is valve. Between left atrium and left ventricle, there is another valve. Okay, the one on the right side is called the tricuspid valve. The one on the left is called the bicuspid valve. Okay, and then from the right ventricle, it's connected to the pulmonary trunk. Okay, there is also valve over here. Okay, that valve is called the pulmonary valve. Okay, on the left ventricle is go to aorta, and there is also valve over here. That valve is called the aortic valve. Okay, so total there are 4 heart valves. Okay, so what is the function? The function is to allow the blood to flow only one direction. So from the right atrium is only going down to the right ventricle. And then from the right ventricle is only going to the pulmonary trunk. Okay. And then from the left atrium is only going down to left ventricle. From the left ventricle is only going to the aorta. Okay, just one direction. From the aorta, go to the whole body and finally go back to the right atrium. From the right atrium, go to the lungs. And from the lungs, going back to the left atrium. So the blood is actually going only one direction. The tricuspid valve control the blood flow between A and B. Okay, so which one? So remember, this is on the right side, right atrium and right ventricle. This is a valve called the tricuspid over here. So between the right atrium and the right ventricle. The bicuspid or mitral valve control the blood flow between blank and blank. Again, if it is hard, the left side, you have left atrium is connected to left ventricle, and there is valve. It's called the bicuspid valve. Okay, it's connected to left atrium and left ventricle. The pulmonary valve controls the blood flow between blank and blank. Okay, so if it is hard and there is right place, okay, so right atrium, right ventricle, this is tricuspid. Okay, and then from the right ventricle go to the pulmonary trunk. There is valve over here. It's called the pulmonary valve. Okay, so this pulmonary valve connects the right ventricle and pulmonary trunk. So right ventricle and pulmonary trunk. The aortic valve controls the blood flow between blank and blank. So it's going to be on the left side of the heart. Okay, you have left atrium going down to left ventricle. There is bicuspid valve over here. Okay, so the blood going down. And then from the left ventricle going to aorta. And there is valve over here. It's called the aortic valve. Okay, so this aortic valve connects the left ventricle and aorta. Left ventricle, aorta. What is the pacemaker of the heart? Okay, so the pacemaker is a group of the heart cell, cardiac muscle that contracted, okay, first without any order from the brain. It is located on the right side of the heart. Okay, so this is group of muscles. It's called the SA node, sinoatrial node. So this is the pacemaker of the heart. It's called the sinoatrial node or SA node. Which of the following statement is true about the hormones? So now is about hormones in your lab experiment. Hormones produced by neuron. No, hormone is not produced by neuron. The one that produced by neuron is called the neurotransmitter. They are produced by the exocrine gland. No, exocrine gland will not produce hormone. Exocrine gland produce liquid like sweat, tears, mucus. Is not hormone. Produced by endocrine gland. That's correct. And circulated in the blood circulation. That's correct. So this one's going to be the true statement about hormones. Produced by the mucus cell. No, mucus cell is exocrine gland over here. So C is the answer. Which of the following statement is true about target cell? So hormone produced by the gland, endocrine gland, is go to the target cell. What is true about target cell? It will have the receptor. Okay, the cell that have receptor for hormone is called the target cell. Which of the following statement about insulin is true? Okay, insulin increase glucose level. No, insulin actually decrease glucose level. So this is incorrect. Insulin decrease glucose level in the cell. Look at this one. That's okay. Look at the next one. Insulin increase the absorption of glucose by liver, muscle, and fat cell. Yes. Insulin decrease the absorption of glucose by liver, muscle. So insulin decrease glucose level in the blood, not in the cell. It should be in the blood. How? By taking this insulin or this glucose and put it in the liver, in the muscle, and in the fat cell. That's actually the process. Decrease the insulin in the blood by putting them or store them in liver, muscle, and fat cell in the form of glycogen. So glycogen is made of many glucoses from the blood. Okay. So this one should be the answer. So the target cell for insulin will be liver, muscle, and fat cell. Which of the following pancreatic cell produce insulin? Okay, so if we see the pancreas, and if we, let's say pancreas is look like this, it's just on the top of our stomach. If we cut this, okay, cross section and see under the microscope, you will see two main areas. The outside area is called the acini cell, and this is produce enzyme. So this is exocrine gland. But in the middle, it's called the islets. It's actually endocrine gland. It's produce hormone. There are 4 hormone produced. Okay, glucagon. Second hormone is called the insulin. The third hormone is somatostatin. And the fourth hormone is called the polypeptide. Okay, so there are 4 types of cell actually in the islets. Yeah, the alpha cell produce the glucagon, and there is another cell is called the beta cell. This is the one that produces insulin. And there is a delta cell, and this is the one that produces somatostatin. And there is F cell, and this F cell produces the pancreatic polypeptide. So going back to the question, which one produces insulin will be the beta cell. So beta cell of pancreas produce insulin. In which of the following conditions fish will swim slow with lethargic movement? Okay. So which condition actually? This is when the fish does not have enough glucose. Okay, the same like persons, if we don't have enough glucose, we're gonna feel lethargic, weaker. And that condition is called the hypo. Hypo means lower, glycemia is sugar. So hypoglycemia is the conditions with lower sugar concentration in the blood. In which the following condition fish will swim fast and darting movement. So this is the experiment that you perform. If you put glucose or sugar in the water, the fish will swim faster because fish getting more glucose. And that condition, or more sugar, and that condition is called the hyperglycemia. Sometimes kids, when they take a lot of sugar, they become hyper. That hyper is actually hyperglycemia. In which of the following conditions fish will swim with regular and smooth movement? Of course, when the sugar is in the normal condition. It's called the normoglycemia. Okay, I think that's all for this lab experiment or lab exercise lab exam number 2. And good luck with your lab exam number 2. Goodbye.